Let's go over the classes of immunoglobulins. We begin with IgG. IgG makes up the greatest concentration in serum, about 3/4 of the total antibody pool. There are 4 subclasses: IgG1, IgG2, IgG3, and IgG4. All activate complement except IgG4, with IgG3 being the most efficient. IgG can cross the placenta, specifically subclasses 1, 3, and 4. It is evenly distributed between the blood and tissues. IgG plays a major role in neutralizing toxins. Subclasses IgG3 and IgG4 are often associated with autoimmune diseases. Next is IgM. IgM is the largest antibody, existing as a pentamer, 5 monomers linked by a J chain. It has 10 functional binding sites, making it the best at fixing complement. It is the first antibody produced in response to an infection and is the marker of an acute infection. IgM makes up about 5 to 10% of the total antibody concentration. It is also the first antibody to appear in the fetus and newborn. Moving on to IgA. IgA is the predominant antibody found in secretions such as tears, saliva, and nasal mucosa. In serum, IgA is a monomer, but in secretions, it is a dimer held together by a J chain. IgA provides the primary defense against infections at mucosal surfaces. It exists in 2 subclasses, IgA1 and IgA2. Now let's consider IgD. The exact function of IgD is still not fully understood. However, it is present on the surface of mature B cells, where it participates in their activation. Next, IgE. IgE is central to type 1 hypersensitivity reactions. When activated, it binds to mast cells, leading to histamine release and allergic responses such as asthma, hay fever, or anaphylaxis. Finally, let's discuss the evaluation of T and B cells. Monoclonal antibodies are used to identify specific surface markers, known as CD markers, on T and B cells. Techniques such as flow cytometry, ELISA, and immunofluorescence are used. These monoclonal antibodies are produced by immunizing a mouse with an antigen, then fusing its spleen cells with myeloma cells to form a hybridoma. Hybridomas are capable of producing monoclonal antibodies indefinitely. Additionally, enzyme immunoassays can also be used to evaluate immune cell function. Let's review the differences between T cells, B cells, and null cells, also known as natural killer cells. T cells are small lymphocytes and make up about 80% of circulating lymphoid cells. Their surface receptors are called T cell receptors, either type 1 or type 2. Their markers include CD2, CD3, CD4, CD8, and CD25. CD4 helper cells release cytokines and can become memory cells. CD8 cytotoxic cells destroy abnormal cells. CD4 and CD25 regulatory cells suppress immune responses. Overall, T cells provide protection against viral infections and tumors, play an important role in delayed hypersensitivity, and help regulate self-antigen responses. B cells are also small lymphocytes, making up about 20% of circulating lymphoid cells. They express surface immunoglobulins, such as IgD or IgM, along with complement receptors. Their surface markers include CD19, CD20, and CD21. B cells evolve into plasma cells that secrete antibodies, or they can become memory cells. They provide immune protection by producing antibodies that neutralize toxins, activate complement, and act as opsonins. Null cells, including natural killer cells, are small lymphocytes that account for about 5 to 15% of circulating lymphoid cells. They carry surface immunoglobulins and complement receptors and may share markers like CD19, CD20, and CD21. Their primary function is natural cytotoxic activity, targeting tumor cells and virus-infected cells. They also play a role in antibody-dependent cellular cytotoxicity, becoming memory-like cells in some cases. In summary, T cells are the main players in cellular immunity. B cells dominate humoral immunity through antibody production, and null cells provide innate cytotoxic defense against abnormal cells. Let's go over the complement system. The complement system is made up of about 21 chemically distinct proteins. Of these, 14 act as effector proteins, while 7 function as control proteins. Together, these proteins play a key role in regulating and controlling inflammation. Now let's talk about the functions. Complement helps activate phagocytes through a process called chemotaxis. One of the most powerful chemotactic agents is component C5a. Complement also participates in opsonization. This means it coats foreign organisms, making them easier for neutrophils and monocytes to recognize and bind. The major opsonin in this process is C3b. The complement system works through cascades or pathways, and both calcium and magnesium are required for these pathways to function. There are 2 main pathways to know: the classical pathway and the alternative pathway. Finally, let's review the control proteins that keep the complement system in check. These include C1 esterase inhibitor, C4 binding protein, factor Y, which degrades C3b, and factor H, which competes with factor B. In summary, the complement system is a highly regulated cascade of proteins that activates inflammation, enhances phagocytosis, and protects the body against pathogens while being tightly controlled by specific proteins.